Hello everyone and welcome to Instant Biology by Dr. Neela. In the previous classes we have discussed about what is development, and in today's lecture we would be talking about what is or what are stem cells. You might be knowing that we have started a new series on developmental biology. The first lecture, as I just said, was on what is development. In this lecture I would be talking about what are stem cells and what are their functionalities and how do stem cells divide. So if you want to understand this, so stay with me throughout this lecture. I'm sure that at the end of the lecture you would be gaining a lot of things. Now for those students who understand Hindi in a better way, so I have already uploaded the video on Hindi in my YouTube channel in the developmental biology playlist, so you can check that out in Hindi. But today's lecture would be in English. One more important thing, that the notes related to this lecture have already been posted on the Telegram group. So if you are interested in the notes for studying, you can check out that PDF file on the Telegram group. And the link to the Telegram group is also in the description box of this video. Okay, so let us start without wasting much time. So what are stem cells and how do they divide? What are stem cells? The first question that comes to the mind is, what are stem cells? So stem cells are some special types of cells that can recreate themselves. That means they can form another cell like them. Okay, so what does recreation means? Re and creation, these are this recreate term is made up of two words, re and create. Re means again, create means form, that means form again. So stem cells have the capacity to form a cell like themselves again. So this is the functionality, this is the basic concept about stem cells. Okay, so what are stems, or what can stem cells do? So stem cells can generate a progeny, that means they can also form a cell that later in the stages of life can differentiate. What do you mean by differentiate? Differentiate means to understand or to gain a particular function, right? So let me give you an example. Suppose a student is studying in class 10th or 11th or 12th. Now, as of now, he or she does not know about the future or which science or which stream he is going to take. I'm talking about the Indian education system. So after a student passes from class 12th, he would be deciding that yes, I am going to take science stream or commerce stream or engineering stream, right? So a student, I can take an analogy, a student who is in class 12th is in the undifferentiated stage. That means he does not know his or her own function or what he or she is going to become later on in the life. So we can say that the students of class 12th are undifferentiated. The same analogy can be applied to a progeny or a daughter cell that is formed from stem cell during the division, and this daughter cell will later on go on to understand or to gain a particular function, and this is called as differentiation, and the cell will be called as differentiated cell. For instance, this daughter cell becomes a lung cell, or it can become a liver cell, or it can become a heart cell. For the matter of fact, it will just gain a particular function. Before that, it was not having any function, it could have become any other thing, right? So I believe you would have understood what does differentiated cell means. So the first point is the stem cell can recreate itself, can reform itself, can again build its own self, and the other thing is while division it can also form a daughter cell that can be differentiated in the later stages. Now another thing is, another point related to stem cell is these are undifferentiated cells. Now whenever or wherever you might have read, you would definitely have read this statement that stem cells are undifferentiated cells. What does undifferentiated means? Undifferentiated means that they do not have any function. As I just said, that undifferentiated cells do not have any function. So stem cell do not have their any function, but when they divide, they can form a cell of their own kind as well as a cell that has later on the tendency to become a differentiated cell, right? So the third point is the stem cells are undifferentiated cells, and one more interesting feature about stem cell is that they can proliferate, or they have the properties to continuously multiply. They can continuously divide or multiply, whatever you say, because in biology division and multiplication means the same thing in terms of cells. So the cell or the stem cell will have a proliferative property, or it can continuously go on dividing. So these are the three features of stem cell. First, again, I'm just revising for you. First feature is that stem cells can recreate themselves. The second feature is while division, it can form a daughter cell that will be later on differentiated. The third point is that it has a feature of being undifferentiated. It will stay as undifferentiated, and later and plus it has a property of proliferation. That means it will continuously go and divide. So these are some of the features. This is what is stem cell. These are the features of the stem cell. So let us move on to the next slide. Now you cannot point out and say yes, there is just one type of stem cells. There are multiple types of stem cells. We would be discussing all these stem cells in great detail in the upcoming lectures. For example, there are totipotent cells, there are unipotent cells, there are multipotent cells, there are pluripotent cells. So stem cells can be of n number of types, but all the different types of stem cells will have one function. Will have two properties. One is proliferation, that means they will be dividing, and the second feature is they would be later on getting differentiated. Okay, both of these features are very useful in the modern medicine. Why am I saying it is useful in the modern medicine? Suppose some individual contracts some kind of a disease in which the liver or the lung or maybe the heart is damaged. Now, as of the current scenario, you just have a replacement, right? You will have to replace the part of the liver, or you will have to replace the part of any organ that for the matter of fact is damaged. But when the stem cell concept, or when the stem cell studies would be on a greater stage, then you would realize that you can plug in certain stem cells at the site of the damage, and these stem cells will gain the function, will differentiate, will proliferate, and later on they will form the damaged portion of that organ. You will not have to worry about replacing or transplanting that organ from some other organism. Right? So this is the potential, or this is the height of the development that stem cells can achieve. Right? So the first point we have cleared, that different types of there can be different types of cells. I told you about certain types of stem cells, but all in all they will have two properties, proliferation and differentiation, plus they have a lot of application in the modern medicine. Now, where do stem cells reside? The other question that comes to mind is, where do stem cells reside? Where is the location of the stem cells? So stem cells, we can safely say that stem cells reside in the stem cell niche. Now what do you understand by niche? Niche is basically an idea. It's not an habitat. If it is a term that is related to ecology, it is not an habitat, it is an idea. That means it comprises of the functionality also, right? So stem cell niche basically refers to the area where the stem cells will reside, but the stem cell niche will have an overall effect on the stem cell, that it will create or it will maintain the stemness of the stem cell. What do you mean by stemness of the stem cell? That means when the stem cell is present inside the niche, or inside the stem cell niche, it will continue to remain as stem cell. That means it will continue to remain as undifferentiated, and it will continue to proliferate. This is the functionality of the stem cell niche. Now how does that happen? How does the stem cell niche make such kind of arrangement? The concept behind this is, stem cell niche basically has certain kinds of paracrine factors or paracrine signals. Now these paracrine signals or paracrine factors will lead to maintenance of the stemness of the stem cells, or maintaining them in the undifferentiated state, or maintaining them in the continuous dividing phase. This is the concept. So what have I written over here is, stem cell niche is a micro environment. Understood that it is a very small environment, and it has local as well as long range signals. What do these signals do? Again, quiescence, division and differentiation. Understood? That means when the stem cell is present inside the stem cell niche, it will have these kind of things. But if the stem cell leaves the stem cell niche, when the stem cell will leave the stem cell niche, then there is a probability that the stem cell niche might not have that much effect on this stem cell, and it will start differentiating, it will start doing its own thing, or it will start behaving as any other kind of a cell, or it will gain any function. Understood? So In the stem cell niche, continuous division, undifferentiated thing and quiescence, all of these things have to be maintained. I believe you would have understood this. Let us move on to the next feature. So stem cells have the power or potency to produce different types of cells. This is what differentiation means, right? So stem cell will later on, or it can later on, produce different types of cells. Understood? Therefore, totipotent cell, if I take the example of a zygote, right? If I take the example of a zygote, what is a zygote? Embryo, basically zygote is forming from the fusion of the male and the female gamete, and this zygote has the potency to form the entire organism. That means it can, or zygote has the potency to form any cell. That means zygote is behaving like a stem cell, right? So stem cell division and self renewal. Now we are moving towards the next topic, and that is basically how does a stem cell divide and how does a stem cell renew itself. Now the division, if we want to study the division of the stem cell, it can be basically of three types. What are the three types? One is single cell asymmetry. The next one is population asymmetry. And this population asymmetry is also called as symmetrical differentiation. And the third one is adult stem cell lineage. Now we are going to study all of these three in the upcoming slides. Let us start with the first one, that is single cell asymmetry. Now what do you understand by single cell asymmetry? Now asymmetry means the formation of something that are the formation of two things that are not equal, that are not symmetrical, right? So single cell asymmetry means that when a particular stem cell will divide, it will produce two cells, but these two cells will not be the same. What do I mean that they will not be the same? If I say that particular stem cell divides like this, suppose this is a stem cell and this stem cell decides to divide, it is going to divide in this way, and one cell will be stem cell itself. What did I say? That stem cell have the property to recreate themselves, right? So this was a stem cell. Stem cell divides and forms another stem cell, but again the second cell that was formed is a committed cell. Committed cell, I am writing CC for committed cell. What do I mean by committed cell? Now this cell will be committed for getting differentiated. That means this cell will later on go on to gain its feature. Now this can be any cell, for example lung, for example liver, for example heart. It can be any cell. So how has the stem cell divided? It has divided in an asymmetrical fashion. One cell that was formed is the stem cell, and the other cell that is formed is the differentially committed cell. One more important thing is that in this particular way, or in this way, the stem cell even after dividing, so it is divided, then also it is maintaining its number, right? Over here one cell has been deleted because one cell has divided, and again one cell has been produced. So +1 and -1, the number of stem cells in the stem cell niche remains constant in case of single cell asymmetry. So this is a diagram of Gilbert only. Whatever I am teaching is from Gilbert. You can just take it as a cross reference. So this is a single cell symmetry. You can see that the stem cells, when it divides, this arrow basically shows you that it is forming its own type, and this arrow shows that it is forming a committed cell also. This is called as single cell asymmetry. Understood? Now let us move on to the next slide, and this is called as population asymmetry. What do you mean by population asymmetry or symmetrical differentiation? Understand this, that certain stem cells, they will divide and form two cells, and these two cells will be committed. Both of these two cells, or both of these cells will be committed. And certain other stem cells will divide in such a way that both the cells that they form would be behaving as stem cells. This is called as population asymmetry. Why am I calling it asymmetry? Because certain cells behave in such a way that they form both stem cells. Certain cells behave in such a way that they form both differentiated cells. That is why we are calling it as asymmetry. But If I am talking about the other name, it is called as symmetrical differentiation. Why am I calling it symmetrical differentiation? Because one cell will form either both stem or either both committed. So over here you can see in the diagrammatic representation also, suppose this is a stem cell and this stem cell can form both stem cells. This is also stem cell and this is also stem cell. This can be one probability. And in the other case, the stem cell can form two committed cells also. Two stem cells and two committed cells also. So this is what have I written. Some stem cells produce differentiated cells when they divide. Produce differentiated cells when they divide. There are certain stem cells that can produce or that can divide in this way, producing two committed cells. Whereas another set of stem cells will divide in a symmetrical way to maintain the stem cell pool in this population. That means they will divide and produce both the cells as stem cells. Now this is also called as symmetrical renewing or symmetrical differentiation. Now I told you why it is called as symmetrical differentiation. I hope this is clear. So this is the second way in which a stem cell can divide. Let us move on to the next slide, and in this slide you can see that adult stem cell lineage is present. What do you understand by this? Now this happens inside multiple organs. What happens is that a multipotent stem cells. Now what is a multipotent stem cell? Multipotent stem cell is a stem cell that can form numerous different types of cells. Numerous different types of cells. Not all cells, but yes, numerous different types of cells will be formed from multipotent stem cell. Now this multipotent stem cell will lead on to form the committed stem cell. What do you mean by committed stem cell? Again, it is a stem cell, but it will have a limited potency of forming different cells. That means the number of cells that a committed stem cell can form will always be lower than the multipotent stem cell. That means multipotent stem cells can form a huge number of cells, whereas committed stem cells can form some cells only, one or very few cells. Then this committed cell or this committed stem cell will lead on to form the progenitor cell. Now what is this progenitor cell? It is also called as transit amplifying cell. Now this transit amplifying cell after multiple division will form the differentiated cells. Now what is this differentiated cell? This can be liver cell, this can be lung cell, this can be heart cell, and in this way the adult stem cell lineage works. That means starting from multipotent, moving on to committed stem cell, then to progenitor cell, and then to finally differentiated cell. This is how the stem cells take over the different functionalities inside an organ. I hope this is very clear to you. In the diagrammatic representation also we have shown that you can see that this is the multipotent stem cell. This is a committed stem cell. This is a progenitor, and the progenitor continuously divides. You can see like that it is dividing again and again and again, and finally forms a differentiated cell. Right? I hope this is clear. So these are the three ways. The first one was single cell asymmetry. The second one was population asymmetry, and the third one is adult stem cell lineage. It happens inside multiple organs. Now with this, I would like to show you a GIF. You can see that in this GIF, you can see that the stem cell can form n number of organs. You can see that it can form heart, it can form epidermis, it can form your hands, it can form your brain, it can form your retina, it can form anything. That is why I was saying that stem cell therapy is basically the future of medical sciences. Sooner or later you would observe that in case of damage or in case of diseased organs, stem cells will be used in order to replace those things. Now also in multiple laboratories, in multiple research facilities, these kind of experiments are going on, and a lot of success has been obtained in this area. So I hope you would have understood this entire lecture. If you have understood this, please like the video, subscribe to the channel, share with your friends, and also do not forget to press the bell icon so that all the updates from this channel reach to you ASAP. Thank you.